Hello, everyone. Welcome back to the second lecture of our 2024 Kansas Distinguished Lecture Series in Statistical Sciences. So for those of you who are new today, my name is Sajina Dang, and I'm an associate professor in the School of Mathematics and Statistics at Carleton University. And I'm also the chair of the nominating committee of the 2024 lectures. So this highly prestigious lecture series is delivered by an exceptional researcher working in the area of statistics and data science. This annual lecture features one general lecture and one more specialized lecture. So this series was launched in 2002. And since then, we have had 22 internationally renowned scientists from around the globe give presentations on their impressive research. So the candidates are nominated annually by a panel of 6 researchers from universities across Ontario. This year, the nominating committee comprised of Dr. Aisha Ali from University of Guelph, Dr. Jessica Brunsfeld from University of Toronto, Dr. David Haziza from University of Ottawa, Dr. Kelly Ramsey from York University, Dr. Michael Wallace from University of Waterloo, and myself. So today it is our pleasure to have Dr. Susan Holmes here today. She is a professor in statistics department at Stanford University and a highly accomplished researcher. Her research interests include statistics, reproducible research, data science, and statistical application in microbiology and immunology. She has published over 140 publications, including conference proceedings, 12 chapters and reviews, and book titled Modern Statistics for Modern Biology, which is a great resource for aspiring biostatisticians. Among the many honors and awards received by Dr. Holmes include Bioconductor Community Award, IMS Medallion Lecturer, the Britton Lecturer, the Bremen Lecturer, and the NIH Transformative Research Award. Furthermore, she's also a fellow of the Fields Institute and a fellow of the International Institute of Mathematical Statistics. She is also very active in statistics and bioinformatics community. Among her many accomplishments, I'm only highlighting a few. She is currently an associate editor for the Journal of Open Source Software. In the past, she has served as an associate editor for many journals, including Journal of Statistical Science, Annals of Applied Statistics, Journal of American Statistical Association, and Journal of Statistical Software. She has also served on the scientific committee of the Women's in Data Science at Stanford, the Science Board of Sydney Mathematical Research Institute, and a panelist for NIH. And today you will be hearing the second part of our second talk for this seminar, for this lecture series. And the talk is titled Statistics and Geometry for Heterogeneous Data. So without any further ado, let's welcome Dr. Holmes.
So I'm going to, I hope that it's going to stop talking at me if that's cool. This is a sort of more specialized lecture. Yesterday I gave a motivating lecture about why the microbiome data is difficult to handle. And I want to talk a little bit about the interface, more mathematical than yesterday, which is with geometry and how geometry comes into a lot of the things that we try to do. And so I use the word embeddings for many things, and I think that yesterday I talked about language models for the microbiome and how topics were useful. And we know that the large language models that have been developed, the second step after you make the tokens and the topics, are the embeddings. And it's the same for biological data. That is, embeddings of very high dimensional data into lower dimensions are really important. And so I'm going to talk a little bit about the challenges, the state spaces, and in fact, the underlying latent manifolds that we're trying to estimate. Some of the data, whether you are using uncertainty quantification in a Bayesian framework or whether you're using the bootstrap, there's a challenge that comes with what do you do with the posterior distributions on multivariate data? That is, they can only be useful to biologists if they understand or can interpret the output. So that's going to be part of it, and I'll show some examples. And at the end, I'll finish with some open problems, which I think that statisticians could bring a lot to bear on. So the real world data that we look at, not only they're not uniform, they're never uniformly distributed, they're never independent. They're never identically distributed either. So in biology, we don't have the standard statistics framework of IID data where you have to do mixtures and you have to do hierarchical models to capture some of the differences in the densities. The features, as I showed you yesterday, they're not predefined. And so often the potential dimension could be infinite. And so you have to take that into account, and dimension reduction becomes very important. And then, of course, as in the case yesterday, we have documents of different length or samples of different depths of reads. So you have this heteroskedasticity that you have to deal with. And so that's also something that we want to be able to look at. Brett Efron always says, statistics is much more difficult than mathematics, and his idea there is that, you know, in mathematics there are numbers and there are functions. Probabilities are functions on functions. But statistics, it's functions of functions of functions, and they're computed from numbers. So you can see that actually statistics is quite a difficult subject. And sometimes people simplify by saying, okay, we have a manifold, for instance, but we're going to suppose that all the data are uniform on that manifold. That's rarely the case. So there are many unknowns, and sometimes there's a conflict in the unknowns, and we have this problem of unidentifiability. But we want to find sort of small, low-dimensional approximations for the support of the probability distributions. And we want to find out, you know, what these distributions are, which they are never uniform. So a starting point to think about embeddings is an old starting point from a paper by Schoenberg, in which he addressed the question of, you're giving a matrix of positive numbers, under what conditions is that a matrix of distances between points? So can you make a Euclidean embedding such that this matrix, this distance matrix, is the distances of the points in this some-dimensional Euclidean space? And that's the basis, this approximation is the basis of what we call multidimensional scaling, and in ecology it's called ordination. And if you start with the original data, then you have to, you know, we center the data, and then we compute a cross-product matrix. And if you center on the left and the right and take the square of the distance, then you get this matrix B, which is equal to this cross-product x prime. So Schoenberg's result was an exact one. He gave the condition under which, you know, if B is semi-definite positive, then you have this distance. And then a few 20 years later, Eckhart and Young showed that you could have an approximation. That is, it gave a good approximation, and this is done doing singular value decomposition in the same way we do it when we're doing PCA, but using the distances. And so you have the singular values, if you write them down like this, you know, for rank R. Here, there are singular values which are non-zero, and the eigen decomposition of XX prime is the, you know, the square of that. And it enables you to go from a distance to a space underlying it, where the distances between the points are representative of that distance matrix. And this is an ordination method. In ecology, we call it PCOA, and it's really important in the study of the microbiome in many cases, where you don't have the original data, but you just have distances. So this is the algorithm, and so that's the first embedding in some sense. It's one of the embeddings that was embraced by the econometricians in the 1950s for trying to understand the space of preferences. So embeddings have this long history, and they are very useful. That is, they enable to see as if there's an underlying dimension which is low. That is, you start off with a lot of data with many measurements, but it might be that actually a natural embedding is only 4 or 5 dimensions. So I use this, the very non-Euclidean data, in that you have, for instance, phylogenetic trees, and you can define the distance between phylogenetic trees by a nearest neighbor interchange. And these distances are continuous. That is, if you consider that I went from here to here by making this edge smaller and smaller and smaller, and so you can measure how far they are as a continuum. And so this gives a space of trees where you can compute the distance between 2 trees. So for instance, here we have 2 phylogenetic trees as they sit in this tree space. And if you go by the tree, which is all without any edges at all, which is what we call the star tree, you can always shrink all the edges to 0 and then grow them. So you can always compute distances, and it turns out that this is what we call a cat 0 space. So it has negative curvature. The picture here is not completely right because the curvature isn't perfectly covered. It's like little segments. But the idea that you have a rectangle which is thin and thinner than a Euclidean one is the idea, and it's sort of like a hyperbolic space. So this gives us properties, and this is why Markov chain Monte Carlo works well on tree space, and it's why we can compute averages of trees. We have a notion of variance on trees, so we can do statistics on trees. And one of the things that I was interested in was we're using the bootstrap, and we bootstrapped the data in order to see how variable the trees were. And we wanted to make the equivalent of confidence regions, but for trees in tree space. It turns out that's very hard to do. And using an embedding is much more natural. So you can compute distances between trees and then use that. So the paradigm behind the bootstrap is that if you were given many, many data sets, then you could look at all the estimates from the different data sets that came from the same distribution and see how much they vary and look at what we call the sampling distribution. And the bootstrap cheats, that is that instead of having the actual data with the true distribution, we have a fake approximation which is given by the empirical distribution. And that's how the bootstrap works. And here we put little stars in here to say that this isn't a true sampling distribution, it's a bootstrap distribution. The question comes up then, where is a region where 95% of the trees lie? And so just to show you an example, here we did the multidimensional scaling of the bootstrap trees, and we got, it's probably sufficient to take 2 dimensions here. And what we see is for these many trees, they're actually clusters, and we could go in and find out in each of these bootstrap, which were the observations that were bootstrapped out, which made these changes. So we can actually see these discrete clusters. So in this case, we have a way of doing the bootstrap and finding confidence regions for the trees in a non-Euclidean space. So it's a non-Euclidean geometry, but we cheated because we embedded it in that way. So that's distances and how we can use distances between trees to do some kind of uncertainty quantification. And now the next step is many times the data themselves, they sit on a low dimensional manifold. So I'm thinking of things like in biology, there's something called Waddington's developmental landscape. And so it's a picture with a manifold, and we have the data sit there and there are local minima. And in genetics, you have the fitness landscape. You have the idea of a low dimensional manifold, And you don't see that. That's a latent, hidden, important support of the data, but you don't know it. And it might be that your actual data are these red points here, that is, and they have a manifold, so the points are actually on the manifold, but there's measurement noise. So you might have something like this. So the task that you want to do is, where's the manifold? We want to estimate the manifold and then the projection of the points on that manifold. And I'm going to show you a very simple example. This is political data, and it comes from voting, and it's in the House of Representatives in the US, and it's roll call. And the data are, actually it's yay, nay, or abstain. So it's -1, +1, or 0. And what we did here is we did a multidimensional scaling with a certain distance of this data. And then we look at a 3-dimensional embedding, and we see pretty clearly that there are two groups. So this is the original data. This is what it looks like. And there were about 400 representatives in this case. And, about 670 votes for that year. So it's high dimensional. But in fact, you could represent it. Most of the information is contained in 3 dimensions. So you have this low dimensional representation. And what's more important is you can see that the data is sitting on two arcs. We call them horseshoes. And of course, you know, if you color the data, I don't tell the program, you know, to start with the Democrats and the Republicans, but I can just color the points of the representatives according to their party. And you see you have the blue and the red. You know, the Democrats are here and the Republicans are here. And so you obtain two very clear manifolds. That's what I call a manifold in that case. And there's a meaning, in some sense, to having arcs like that. That means we have a gradient. And the gradient is actually, you can explain the gradient by, you know, the more to the left, the more to the right they are. And just to show you what we did actually transform the data a little bit, because I used a L1 distance, but then I took a kernel, did transformations, and then renormalized, and then you compute the, you know, the eigenfunctions. And it turns out, there's a paper that I wrote with Percy Diaconis and Sharad Goel, that we can actually compute completely the eigenfunctions, what they are. We have trigonometric eigenfunctions in this case. And the physicists knew this. After we wrote the paper, we realized that this is something that physicists know in random. They have what they call Euclidean random matrix theory, and they also have the eigenvalues and the eigenvectors. And I'm not going to go too much into that. But the distribution tells us, in some sense, the dimension of the manifold. So here, these are the first 3 eigenfunctions that we have, F1, F2, F3, and this is what they look like when you put them together, F2 and F3. This is what it looks like. That is, you have this horseshoe. So we can make a model, a physics model. If the points are just along an interval, and if they're uniformly distributed, they would be perfectly along this horseshoe. And the theory tells us this is what the eigenfunctions would look like. So we have a theoretical reason to expect the data that we saw. And this is actually the picture that you get from the model, this generative model that I just made up. So this is 3 dimensions, and we have these 2 arcs, and you know, we have the 2 groups. And one of the things we supposed here, and it worked out very well, is that you have a uniform distribution along the manifold. And the true data, this is what it looked like. These are the true eigenfunctions of the observed data. And so they're very close to the theory, and it works pretty well. And here you can see that the ordering, this is the ordering that's going to tell us the left to the right for the Democrats, for instance. And if we look at a score from the outside, so we bring to bear some other information to try and interpret this manifold, we have something called the National Journal Score, which measures how left-wing or right-wing a Democrat is in particular. You can see that these scores are completely aligned, very correlated with what we get along the multidimensional scaling of this eigenfunction. And you can also see something else, which is the gradient for the Democrats is very different than the gradients for the Republicans. So they have their own gradient. So now I'm going to go a little bit further and see how I can adapt this method to reality. And what I mean by reality are the cases where the data are not uniform along the manifold. So this is a gradient analysis of microbiome data. It's called the Tara Ocean data. And here we have these points, the gray points are all samples. And then we're trying to find the underlying ordering of the samples. And we've projected them in some sense to the closest gradient manifold. And we want to know the uncertainty with which we know this. So we use a Bayesian method. And you can see that the points are not uniform in anything near uniform along the manifold. And we have in particular large areas where there aren't a lot of points. So we need a method which takes into account the uncertainties are different at different points in the curve because of this unknown. Now I want to make a parallel with a method which came out after we did this work, but tackles this problem, I would say better, because it's much more general, but it's exactly the same issue. And that's how UMAP works. So when you're doing manifold learning, or if you're doing topological data analysis or various math based methods for geometrical estimation of the support, you do much better if you have uniform data. And there are ways of lifting the data. So you do a change of measure so that you can make the data more uniform. Now, we do that, we increase the dimension when we use kernels, but you can also do that by lifting things in a sort of differential geometry way and adding more coordinates and having maps here where the data are uniform on the maps and you can compare them on this bigger space. And that's actually how UMAP works. And the U in UMAP comes from that goal of uniformly distributed. And it's quite related. Ours only works in one dimension and UMAP works in many dimensions. You can have a much higher dimensional manifold, but it takes into account the sparsity. That is, from time to time, the radius of connection can be much larger. You allow yourself that. So with Lan Huong, we did a Bayesian unidimensional scaling for trying to find these gradients. And so we looked at distances between points. In our case, it's microbiome data, so we use the Jacquard distance. And there's a true distance along the manifold, and then there's a distance, a noisy distance in the observation space. And in order to tackle this, we have to model the distance's heteroscedasticity. So we do this with a Bayesian model. And so it's something like this, where you have all components and the hyperparameters. And we summarize this model in this way, but it means that you can just use Stan and input the data in a very convenient form. So you have to decide, for instance, the prior on the variances. Is this positive part of the Cauchy, and we use a beta distribution for the taus, which are the points on the curve. So we have the curve, we're going to parameterize it from 0 to 1, so you need a distribution over 0, 1, we use a beta for that. So we model the distance's heteroscedasticity in that way, and we have estimates from the data of what the local standard deviations are. So this is done with a standard formula for the local standard deviations, and then we use Stan in this case to try. We use a variational Bayes, and we use a principal curve as our original initiation, which is pretty simple way of doing it, but it works very well for this data. And these are the hyperparameters. And the output that you see, I made an extra move. So this model enables me to generate many, many data sets from the posterior distribution. And this happens when we do Bayesian factor analysis and many multivariate Bayesian methods. And the question is, how do you go from those many posteriors to something that you can interpret? And so the contours is the quantification, and that actually is not trivial. It goes through a step that we call registration. So we build the uncertainty contours. I use the word registration. You can use other words. Alignment is another word which is quite appropriate. And the way to think about it, is to think about image analysis. And so as some people here know, I write papers from time to time with Percy Diaconis, and we did a study of coin flip. And so we took movies, very many images per second of how ordinary people flip coins. And I say ordinary people because my husband can flip a coin 10 times in a row, all coming up heads. And so ordinary people flip coins, and it doesn't always come up the same way. And we wanted to know, can you tell by looking at pictures, what is it always 50-50? Or if it starts heads, does it have a higher probability to end up heads or not? And so we did a lot of data collection, and this was the first image analysis that I had ever done. And we had marked the coin a little bit, and so this is just a picture from the coin. And in this estimation, here, this blue line is supposed to represent the normal to the coin. And that was important in the physics equations that we were writing out. So when I looked at the data in 3D of the normals to the coins, and these numbers actually mean something. They're the sequence in which the images occur. It looks like a mess. So these points, in some sense, the normal is not uniquely defined. It could be that you take it, the top one or the bottom one. And so that problem actually creates a mess because the data needed to be aligned. So we aligned the data. And if you align the normals, this is what you get. You have this nice arrangement, completely circular, where you have them in order. So here, 87, 88, 89, that's the sequence of the images, and the normals are moving in a continuum like that. So that's a registration. That's what I call registration. It's this alignment. Now, if you're trying to do registration for embeddings, you can do it in various ways. The one that I like is something which is a generalization of the correlation coefficient, which is called the RV coefficient, where you just take a inner product. It's a vector covariance. You take an inner product between either the characterizing kernels or between the distances, and you can normalize them. So this is the Frobenius inner product. And that enables you to see how close the embeddings are. Some people do procrustes, but here I consider it to be symmetric, like correlation coefficient. So I use the RV coefficient. Here's an example of how we've used it. This is a paper that I wrote with Lan on dimension reduction. And the idea here is you want to do PCA, you do bootstraps of PCAs, and you want to see how stable the output are. Well, if you bootstrap data and the eigenvalues are not too far apart, but this is something that you could get. And so you get sort of a mess. That is, here I have bootstrapped 5 samples. And I'm looking at these 5 samples, and these are what would you call the contours or the convex hulls of the 5 coordinates that I get from these 5 bootstrap samples. And the problem that's occurred here is the eigenvector in PCA is not defined up to sign, so you have either the positive or the negative direction. So you have some here, and they're equivalent to some here, so the sign. And then you also have the first and the second eigenvectors, and I have switched if the eigenvalues are not too far from each other. So you don't get something nice, and you need to do more alignment. And you can think about this in a way which is similar to what happens when you do bootstrap of clustering. When you bootstrap clustering, the identities of the clusters themselves, in one bootstrap, you might have this cluster that you've called 1, and in the third bootstrap, that cluster might have labeled 6. So you have to do some alignment between the clusters before you can compare the bootstraps. Here it's the same idea, but you have to do a registration. So how do we do the registration? We compute the RV coefficient between all the bootstraps. So if I have 1,000 bootstrap, I have 1,000 by 1,000 matrix, and then I compute the first eigenvector, and that will give me the best place to align. So we project onto that first eigenspace, and this is how we got these pictures of, how confident you are of a PCA using a bootstrap. So these are ways of using uncertainty quantification, and in order to be interpretable or useful in some way, this overcomes sort of the registration problem. So we did this local variance, so we rescaled the weights, and then we combine the posterior peaks. I'm going to show you a sort of more evolved example that we did, and that's for the microbiome. If you're doing a factor analysis model, this is work that I did with Lorenzo Trippa and Sergio Baccalado and some co-authors, where we did an ordination on certain distances, and it's the same thing. That is, each of these is a sample as it's seen in the ordination. We wanted an uncertainty contour. We wanted to know whether we were sure whether some groups were separated from other groups. And so this is this method. And so in order to do this time I'm not going to use the bootstrap, I'm going to use a full Bayesian non-parametric model. And we're postulating that there's an underlying low-dimensional latent variables for these bacteria. And we have to have dependent microbial distributions. You can't use a multinomial for this, because as I said yesterday, most of the bacteria are dependent on each other. So we have this Bayesian model, and here I have different populations, and then I have different taxa, and I did this Bayesian method 1,000 times, and we used a method which is a kernel-based approach to these approximations, and we postulate these lower dimensions. So we have a fully generative model for generating the posterior, and I'm not going to go into too much of the details. This is a paper that appeared in JASA 6-7 years ago, so that's not, this part isn't new, but what I wanted to show you is the difficulty that we came across, which is very similar to what I showed you for the PCA, is all the Bayesian posteriors, once we projected them, this is what we got. And it's very unsatisfactory. It's very messy. So this is the first output for the Bayesian posteriors. And I say this because there's several people are doing Bayesian factor analysis, and this problem that you have to do this extra alignment seems to have escaped the psyche of some of the people. And I think it's useful. That is, I think that in this case, we did again the registration, and I just want to show you the output. So instead of that mess, this is the output that we got. So what do you see? Well, here in particular, we have these contours of points, which are very small, which means that we're actually very sure about where these projections are. This is a very good latent factor model for most of the samples. So here we have most of the samples, and they separate out into groups. We have 4B, this group, which is sort of purple, is here, and then this other group 4A is here. And there are just a couple of these samples which are very bad. That is, in the sense of the contour of confidence region is very extensive. It covers a large area. And we went back and looked at those samples, and those were the samples which had very low depth, very few reads. So of course you say, oh yes, of course, that's why. But these uncertainty contours have been very useful in experimental design. What's the next good measurement that you can do? Well, you want to do the measurements where you have the most uncertainty. So in an adaptive design, this is very useful to be able to see the uncertainties. So as I said, the densities are unequal. And sometimes we concentrate on the fact that we're looking at distances, and we think that distances are enough, but we know we need the probability distance. So we need to take that into account. So let me finish off with some of the difficulties or some of the challenges or where we are going, trying to understand this. One is, so when I arrived at Stanford, we were running a group because I was doing the tree work with topologists, which was called a computational topology group. And with Gunnar Carlson, he was trying to do what he called topological data analysis. And the way he does it, and the way it's done by mathematician, is you take an interesting topological structure, you have a torus, and you put points at random uniformly on that structure, always. Mathematicians always uniformly on that. And then once you have data, then you're going to try and connect. You can't connect the points are isolated, so you fatten up the points with a radius like this. So here you keep fattening them up. So here at this radius, you get points which are connected, and so there's a certain number of connected components. Here it's the same number of points, here it's a smaller number of points, and then here you start to have points which overlap with 3, and then you have a triangle, and fill this in, so you get what we call a simplicial complex. And as the radius grows, you get the number of connected components and the number of holes, which are called Betty numbers, that you can estimate from the data. So that's what topological data analysis does. And what Gunnar wanted us to do was validate using a generative model. So you take points on the torus and you run the algorithms and you check that you get the right Betty numbers. And unfortunately, mathematicians don't understand so well about probability distributions. And if you take the definition of a uniform by the definition of taking the uniform on one coordinate and the uniform on the other, so for instance, the uniform on the angles and the uniform on the radii, you won't actually get the uniform distribution on the torus, because it's not taking into account the local volume. So you need to do a change of measure. And so you need a correction to the naive method. And you do that by fattening up locally and taking into account the volumes. Now, that taught us something, that is, you can do this, and we use it as a method for generating points. So these are points which are actually generated uniformly on the torus. And so what you see is the projection of the points, it's much denser in some of the points. So if you have a manifold or you have a torus, you're going to have the impression of denser points where you have higher curvature. And then there's a problem, because if you're given, so for instance, in UMath, you're told, start by doing a PCA on the data to bring down the dimensions. So if you start by projecting the data start off on 10,000 dimensions, you should put them down in 25 dimensions. And so you've projected them down. And then you have an unidentifiability problem. That is, you don't know whether you're seeing high density of points because there are a lot of points, because there's a big density there, or because there's high curvature. So you could imagine situations where, for instance, if the underlying manifold is unknown and you don't know the dimension or the curvature, you can't distinguish between changes in density and changes in curvature. So this is my trying to make a picture about that. That is, here, this is supposed to be areas where you have sort of a higher density, but this higher density could come from the fact that you have uniform data on very highly curved points, or it could be that just the data are on a very flat surface and they're very clustered. So this is a challenge. That is, this is for me one of the most interesting open problems currently in high dimensional statistics. That is, we actually don't know how UMaP works, for instance, for doing this manifold, for interpreting the manifolds. And we know that TC does a very bad job. That is, there's been enough simulations and generative models where we show that things are completely unidentifiable. That is, TC will make great big blobs just because you have a lot of points, and they can be very narrow, very small variants, but it'll make a big blob. And there's an unidentifiability which is well documented for TC. But UMaP, we don't really understand how to interpret the changes in density compared to the changes in how curved the underlying manifold is. So that's a sort of challenge, I think, currently, because in a lot of the work I do currently, we do a lot of UMaP. That is, my immunologist, that's their favorite picture to make. And not only there's the question of, in PCA at least, you can look at the eigenvalues and you can see what percentage of the variability has been explained. In TC, it's just a religion that you make square plots with 2 dimensions. And I very often make them go back and take out those first 2 dimensions from the data and redo it so that we can look at the 3rd or the 4th dimension, because sometimes there's 4-dimensional structure. But I think that's a sort of interesting open problem. And this was a lecture for the specialists. And I think that, going forward, it would be nice if statisticians could make tools that were less black boxy, I'd say, than what goes on right now. But so that was the open problem. Thank you.
Thanks for a very interesting lecture. So any questions?
Yes.
Yeah, okay. My question is, how is the head of the math department and Phil President, how are they using artificial intelligence in their math programs such as trigonometry and geometry and all that? How are they using AI in their math programs to help their math students, whether they're undergrad or doing master's degrees or PhDs in mathematics? How is AI?
Well, I don't know about the math department, but in my courses, my ChatGPT completely changed the way I teach statistics to biologists. And the reason is we have tools within, I use our studio. We used to have things like auto-completion, where things are, you start typing a function in R and it'll suggest the, now the method is you write a comment saying, I need a function that is going to input a matrix and my parameter lambda, and I want to find the splines that do, so you write a 2-line description of the function that you want, and then the auto-completion is ChatGPT just fills it in. So it's Copilot, and it's right about 80% is right. And so it's a very good learning tool, because instead of being stuck by what's the name of the function, what's the name of the package, what should I, there's a starting point which is much easier to work with. On the other hand, ChatGPT hallucinates, and so it very often says that there's a package that does just what you need, and you look up the package and it doesn't exist. And so then you have to say, you just say back, the package doesn't exist, and so, oh, so sorry, back, giving you another tool, but, and so you have to do a lot of checking. But I found that the biologists are very happy to have this idea of auto-completion. I must say there's a second thing that I have to say about teaching with ChatGPT, and that is that we realized then that the words, the buzzwords that we use are super useful. And if I can come back to my embedding idea, so large language models, the second step after code tokenization and so on is it creates embeddings, and the quality of the response you get have to do with how local you are in the embedding of the right neighborhood. And so if you've used statisticians' buzzwords, like the bootstrap, like confidence region, like the word probability, you're in the part of the embedding is very relevant to you. So the answers are much better. So it's a motivation for teaching students the buzzwords, the concepts. What are the concepts that we use? And so, because you could say the way my kids used to say to me, well, I don't know, don't need to learn any history at all because I have Wikipedia now. And you could say, well, I don't need to know any coding because I have ChatGPT now, but actually you can only get good quality answers if you know the concepts and the buzzwords. So I think that it's a very good teaching tool, but we're still here able to use the buzzwords. Like, for instance, the word unidentifiability, that has a really important meaning to many of the statisticians. And so teaching the students, these are the words that make the responses right in ChatGPT is the most useful. Anyway, so.
Any other questions?
Yeah.
So you mentioned about the non-uniform to uniform distribution, right, in high dimension. Can you say a few words about like computation of it? Like,
Yeah, so what happens here? So this is the picture which corresponds to what goes on in when we're doing UMAP. So this uses differential geometry really, because what you're doing is you're making maps. So in some sense, you have the points in the Euclidean space, and you're going to put them in the differential. There's going to be a differential map, and you're going to increase. And in general, what you need more. You need more degrees of freedom in order to flatten out the points, but it's only locally. So when you're looking for a neighbor, the next step that we do when we do UMAP, it uses also simplicial complexes, is we connect points. And so it's actually quite cheap So it's not expensive. So you have distances between points and you have the same principle that is that we have in topological data analysis. That is, you connect points if they're close together. So you have a little neighborhood. So you only have, it's not too bad. And the original distances that you have, they're already given to you. And so the computation is not too bad. And it's certainly not, it's not the barrier for me in using UMAP. The barrier is the interpretation.
Thank you. Any other questions?
Yeah, so following up on your embedding thing, I have seen someone transforming data into image first, and then using deep learning to analyze or classify the image. Have you come across that? Or?
Yeah, so what can I say? I don't believe in that because we already do that so badly. So just to explain what I mean by that is I studied spatial point processes for a biopsy in cancer. And so we have points which are cancer cells and points which are immune cells. And then when I make the point process and I show that to my biologist, they immediately say, but there's a moon there, and then there's this cloud thing, and so they will do that. And so the image, that's exactly the opposite of what I want. That's what we call pareidolia. And it's seeing patterns in noise. And so I don't think that but maybe if there's a very good database, that is, if there were a very good reference database of images of data like what I showed you, we have two horseshoes or the explanation of one horseshoe or clusters which are like that, maybe that can be done. But I haven't seen that. I have the opposite reaction because I feel that we overinterpret sometimes and I have long discussions with my biology collaborators about what they see in their UMAPs. Because one of the problems is you have, the separation between the clusters can't be interpreted. That is, locally, you can see what's going on in one cluster, but you can see that cluster is separated from the out. But the layout of how that's done afterwards is not necessarily representative of the true distances between the big. So it's valid locally, but not globally. But I think there's still a challenge in training, maybe training the machine to read all the UMAPs or something like that. That could be a good idea. I do think there are a lot of challenges with still visualization, how popular it is, and how efficient in separating cell types and things like that, but how difficult it is for as a statistician to understand what's the distance and what's the probability density differences, how can I deconvolve that? That's the problem that I think is a really interesting one.
Thank you. And just in the interest of time, if there's one last question, we can take it.
To that point and to align with what you mentioned. So do you think that can be data specific, for example, on omics data, specifically on radiomics? I've tested this. It's unpublished. I didn't get a chance to publish, but first we extract the omics features, and then we convert it to image. And now classifiers perform very well, probably because that's the nature of omics. There's a lot of correlations within the data. That's the structure of the
So it's low dimensional, in fact, right? The underlying. No, I haven't tried it because I find images very hard to manipulate and featureize. So, but you could let the computer do it.
And if I may ask a very quick question.
Yeah.
So can you comment on two things? How would the concepts of unseen data and perhaps longitudinal studies changed these methods. For example, UMAP for longitudinal data.
So longitudinal isn't a problem, you're just adding time, so you have one more dimension. And for instance, when I do the uncertainty, well, when I'm doing registration, and I have a data cube, but my dimension is the bootstrap, but I can have another dimension and it can be time. So I can still find the consensus on which to project all the time points, so you see the trajectory. So I don't have a difficulty with that. For unseen data, so if you say missing data, I don't know, but you know, data imputation, we can do some if it's sparse, but otherwise we can't make up data, right? I don't have an answer.
Thank you. We will take one last question from online. So the question is from Ahmed A. I'm curious about any methods or intuition from Dr. Holmes about handling sparse data sets in the sense of high missingness. These data sets usually stump other methods.
Yeah, so sparse, for me, sparse is not necessarily, so high missingness, I don't think, I don't have very much to say about that. There's just a lot of data missing and you don't, so the only way of dealing with sparseness is to be a Bayesian and have some prior information. Otherwise you have to own up that you can't fill in something. So I think that, you know, sparseness, that's why it's a motivation for having good priors from other studies. And empirical Bayes also works for that. But so I think that's very challenging, the sparse. But there's also sparse where you have zeros, so it's unobserved. So you have zero that is not available. That is, you didn't see in single cell data. And I was saying yesterday, when we have a lot of zeros, we put in a mixture model where we say we have a whole bunch of things which were unobserved. We're going to just put in a mixture of what could be in the unobserved and the other, but you need some prior data about the unobserved.
Thank you. So let's thank our speaker one more time.